So, hello everyone. At this moment, 10.5% of all publications in biomedicine use AI or machine learning, up from 5.7% 2 years ago. So if you now think that this is about 5 million articles per year, you can imagine how much at the moment life sciences are changing with AI tools, and toxicology should not stay behind this. I made my slides available. You see here a QR code. I showed on the last slide again, and I also have marked some of the papers. And obviously, this is a team of people behind this, which is working on this. AI is at this moment everywhere. It started really with this kick-off ChatGPT 2 years ago, coming into daily use, and nobody understood why suddenly AI is there. It is very simple. We have a synergy of different exponential growth curves. The data in the world are doubling every 18 months at the moment. So 90% of all data in the world have been generated in the last 3 years. Our computers for more than 60 years are doubling in capacity every second year at half the price, known as Moore's Law. But AI, since 2010, when deep learning was introduced, is doubling every 3 months. So this year's AI is 8 times more powerful than last year's. And this is essentially the difference between, let's say, ChatGPT 4.5 and ChatGPT 3.5. And if you tried out some of these AI tools a year ago and found them gimmicky, try again. Things are changing. I was blessed that 10 years ago, 2 students joined my lab, Tom Lichtefeld and Alex Mertens, as PhD students with machine learning background. And we started to embrace AI. I'm fortunate that they both are, as junior faculty, still working with me. And a lot of the work I refer to has been steered by them. In these 10 years, AI has increased its capacity about 1 billion-fold. Just to give you a feeling, when we started, what it is now. But it was a relatively early start, and this positioned us to really assume some roles. For example, I'm the field chief editor, the founding field chief editor of Frontiers in Artificial Intelligence, a journal which published over the last 6 years more than 1,000 articles. I'm also have been working with the World Economic Forum last year on the top technologies of one of them being AI-facilitated healthcare, and in this year on large language models changing science. I'm putting this out just to give you a feel. There's at the moment an enormous demand on AI and its transformative work in every field. We are here to talk about toxicology, but I would like to show you in 2 slides something which is really an important message, how fast these developments are changing. You see here how different skill levels of humans have been achieved by AI. The blue curve on the very left shows you how the reading of human handwriting was achieved. These are percentiles, so as the lower bar is AI is worse, or is all humans are better than AI, and the upper bar is AI is better than 100% of humans. It took 18 years for handwriting to be deciphered. But to the very right, you see things like coding. Within only a year or 2, AI assumed the competence of humans in coding. In 2022, already 40% of all coding was done by AI. Last year, it was estimated it was 80%, and the big tech companies are laying off coders, something they were looking for desperately still 2 years ago. Yeah? So this shows you something I think really important. How much this is also invading or taking over in science might be illustrated by BioGPT, software which was released in February last year by Microsoft, and it outperformed already then humans in annotating scientific papers. So taking information out of scientific papers is done better by AI. But in contrast to my PhD students, it's not 1 paper per week, I hope, but it is 1 million papers at the time, and it is not, nothing is forgotten. And in the meantime, over the course of last year, the big problems of reading tables and reading scientific figures and diagrams has also been addressed and improved dramatically. So this is what we should expect. I don't want to go into the problems of AI. AI has a lot of value propositions, but it also has a lot of problems, hallucinations, plagiarism, whatever, name it. And this picture, which I had done by DALL-E 3, is a nice illustration. I said, do the A with devil's attributes and the I with angel attributes. But if you give it a closer look, the angel has a red face and the devil's tail. And I left it like this. This was my first prompt because it shows that AI is not perfect. And we have to be in the loop and decide what we take and what we can't take. But the value proposition is simply unbeatable. AI performs at the moment in human intelligence tests stably around 130 IQ points. In some language tests, even at 160. So find some humans who are assisting you at this intelligence level for free. I also think that the value proposition number one is to free us and improve our work-think balance, because it really takes away a lot of the dull things, the reading of lengthy documents, the reporting of standard things, where we simply have some assistance in these things. They have been publishing about the possibilities in toxicology, which is more or less the now, in these three articles, which are open access. One was done with Nicole, who did the beautiful keynote this morning. These articles I would recommend if you would like to see what is the now of AI in this field. They're all available. In short, toxicology, so toxicology based on AI, from my point of view, is simply making big sense with big computers from big data. You don't need AI if you have an Excel sheet. Then descriptive statistics are better. AI only shines if you have much more data and multiple types of data. And toxicology has become really a data-rich science, big data science. We are away from counting dead animals or the number of tumors in an organ, where you don't even need a handheld calculator in the end. We have high content, we have imaging, as we saw very nicely, we have the omics technologies, we have robotized testing of ToxCast and others, we have sensors, we have more and more literature. 1.7 million articles were taken up in PubMed in 2022. 80% describe effects of chemicals, not necessarily as drugs or toxicants, but as inhibitors, as activators, something which is interacting with the biological system. Yeah? We do have more and more data from the internet. We heard from Barry the number of safety data sheets. We found 850,000 in the internet, for example. And there's more and more databases, like Nicole showed in ICE and others, which are curated from legacy studies and various information sources. An enormously rich field which allows us now to use our computers. And the value propositions are enormous. First of all, to find data. This is a very important piece. But also to make sense of this, integrate this very diverse and incomplete type of information to make predictions. But I would first like to show you, before quickly touching on these others, the improvement of reporting, the digital pathology, which is revolutionizing at the moment, how we deal with histopathology and others. It is really a new era here. I want to show you an example how data retrieval looks like. This was a peer review I had to carry out, or I carried out a year ago. The EPA had carried out this work with a contractor. 2 people worked 18 months to bring this together and carried out some type of read across on the information they found on this substance, which was of interest for contaminated Superfund sites. And after I did my peer review, I used first ChatGPT 3.5, which was at the time used by 99% of people because it's the free version, and I found nothing about this substance. Then I used ChatGPT 4, which was at the time used by 1% roughly, which is considered 40% to be 40% more factually correct, but most important, it found quite a few of the sources of data. But it really got interesting as soon as I combined this with the plugins, which gave access to the internet and also to some better retrieval of supporting information. And it really found everything, all of the sources which were in this thing. It also highlighted a study which was found by the EPA but dismissed largely, which I, after rereading, also would give more emphasis like the AI did. It was not just a ChatGPT thing. BART at the time did not perform very well. It has been replaced by others, by Google. Anthropic Claude was at least as good as ChatGPT in finding these things. So it's not just advertising one company. And there's 160 large language models available by last year. However, I was blown away when Perplexity AI found an entire risk assessment, which was carried out by the Risk Institute of Fragrance Materials, which was missed by the EPA colleagues. So complete risk assessment done on the very same substance, not found. So this shows you just how on a silver platter you can retrieve data for whatever work you want to carry out. And this did cost me less than 1 hour to carry out this research. Compare this to 2 people and 18 months. Let's come to predictive work. This is a study I hope I can be doing very quickly because we published this already in 2018. This was an attempt to use AI and to use the similarity of chemicals not to predict a single property, but to predict from 74 input properties the outcome of the 9 most used animal tests in toxicology. So the classical 6-pack, inhalation toxicology, acute and chronic aquatic toxicity. And we started with 10 million structures, out of which 600,000 had data. And we were able to predict in a cross-validation 87% of 190,000 cases where we had a classification done in a regulatory process. 87% correct. The animal experiments of the 6-pack in the same set of data were only 81% reproducible. And they were only 69% sensitive to find the toxic substances in repeat experiments. So 3 experiments, only 2 of them would find the toxic substance on average. And so the inflation came from the non-toxic substances, which were non-toxic throughout the experiments. Yeah? So that's why the accuracy was at 81%, not lower than this. Okay, so that's why we titled this is outperforming reproducibility of animal tests. This work, which was steered by Tom, continued in 2020. We showed that an even earlier version of this was outperforming animals in predicting skin sensitization in humans, not skin sensitization in the animals. We had 506 chemicals which were tested in human patch tests, and it predicted 80% correctly, while the local lymph node assay only 74%. We then tried that we can use this on batch. We took a list of 4,700 chemicals which are relevant to food in the US, either as contaminants or as additives. We have an incredible 4,500 chemicals which are added to the food in the US, out of which 80% don't have public data for risk assessment. So just to give you a flavor of what I'm enjoying there. In minutes, far less than an hour, we did the equivalent of 38,000 animal tests, yeah? Not animals, animal studies, which would cost about $250 million if you found the capacity for this. And again, in a very small validation set, we found 83% correctly predicted in the system. So showing you the potential to run essentially any number of chemicals in such a model. And last year at SOT, we showed the first time that with some additional training, some input of data from REACH and OPERA and others, that we can also reasonably predict cancer risk of reproductive toxicity. So this is by far not optimized, but the 950 chemicals which were registered with data in REACH, or 1152 for reproductive toxicity in REACH, were predicted again with pretty reasonable predictive capacities. So I think there's no limit really in the type of endpoints. The ONTOX project, which is running in the EU, includes both my group and Tom Wichtefeld's spin-off company. As I said, Tom is half-time still with us. And we are doing something really incredible. We are trying to develop a co-pilot for toxicology. We're using 3 test cases, liver, kidney, and the developing brain. And the idea is we enter a formula, press a button, and we get some type of assessment. We'll tell you, I found data, or I found data for these similar type of chemicals, but it is first of all being evaluated for these type of properties, liver, kidney, and the developing brain, with the expertise of the consortium. But in principle, we are developing this for all properties. And what we're doing here is, first of all, we use a number of data inputs. You heard about Distiller. Tom's company InSilica has developed Sysref, which is doing essentially the same like Distiller, but it's for free. Has been used already for 12,000 systematic review projects. But we have now advanced this also to extract information from the identified papers and learn from humans what are good papers to include for your area of interest. So it is extracting now causal relations and which chemicals are in there, which genes are regulated, which enzymes are affected. So this type of information, Sysref is now feeding into our things. The second element is an enormous breakthrough from my point of view. Tom has been programming with his team BioBricks. BioBricks are a one-line import command for an entire database. So this does cost about 2 weeks per database to program, to understand the database and its programming, and then in the end, use this, that it is a one line you can write import PubChem, import ICE, import whatever. And this then happens in minutes. Also very easy to update what is new, get this thing again. In the meantime, he is at around 90 BioBricks. Many of them already available on GitHub, and the publication on this is at the moment under review. To give you a feel, the first one, we call this ChemHarmony, which included only 4 of these databases, already gave us 200 million triplets of a substance, a property, and a result. 200 million, that's really big data. And that's a treasure trove. So very different to the 2018 model where we had 600,000 substances with some data, yeah? 200 million here. And we're also moving forward of not only looking into structural similarity, which is the first 2, this lady and the dog are structurally very similar, but biologically more similar are the 2 dogs, obviously. And in a test run, we found that by also including biological similarity, the performance in biological assays as a measure of similarity, we're adding about 10% accuracy to our model. So we are very have high expectations that this is really adding to it. The data we then feed in 2 things. One is our read-across-based structure-activity relationship type of approach, the predictive model. The other is we are automatically feeding this into AOP networks and physiological maps, that the perturbation of biology is illustrated, but we're also trying to use this to ultimately predict a probability of hazard from these. This integration part is where we are at the moment. And this is a very important concept, probability of hazard. As toxicologists, we like it black and white. Yeah? Toxic, non-toxic, carcinogenic, non-carcinogenic. That's easy. It's a definitive answer. We close the books and we don't need to follow up. What science can deliver is gray. A lot of gray is a lot of uncertainty, depending on the use scenario, the context of use and others. And we should start to familiarize ourselves that this is the reality and that this is what we can communicate and this is what we're working on. The advantage is that no longer will alternatives and new methods be a threat, that they will reclassify every substance. They will only shift the tone of gray or the uncertainty associated with it. This might make them much more acceptable in the future. We have followed up on this concept first with a white paper. What is this? Then we carried out 3 workshops. The first one has already been reported. The second one is at the moment being finalized as a report, and the third one is in writing. So we're really embracing this concept because AI is delivering probabilities. It is not necessarily expressing them, but ultimately it is always looking for the most probable result, for the most probable next word when writing something. And then you can ask the AI, how probable was this? And this is a unique information because it can tell you this is very solid, or it can tell you, yeah, it was borderline. And if you ask, what is it above the threshold? Yes, it was, but it was very close to the threshold. This is a very, very big opportunity here. So in short, my message is probabilistic type of risk assessment becomes only beautiful by AI. Because you get in the data, and AI is also handling the multiple type of information from in silico to in vitro to in vivo to human epidemiological studies. To give you a flavor where we are at the moment, we are training at the moment on a database which has 254 million chemical activities for 117 million chemicals. 4,000 of their properties have more than 1,000 data points, which means we believe that this is covering the chemical universe, at least representatively. And in our training, we see at the moment for 4,000 properties that on average, they're predicted 91% accurately. Imagine, 91% accurately for more than 91% for 2,000 properties of these chemicals. At the moment, we're looking to what are these properties, what are the ones we are predicting well, what are the ones which we are not predicting that well, and we are annotating this and creating a user interface. And by the end of the year, our partners in ONTOX will get access to the model and then they will explore it for their expertise levels. They are identifying data sets which we have not trained on so that we can really find out how well this does. So how are we doing this time?
We could, yeah.
2 minutes. Okay, very quickly. This has a lot of impact for other areas as well as not just for regulatory science. A big thing for us is green toxicology, so front-loading these things. We have been working for the last 10 years on how to predict properties of chemicals, perhaps even from structure, just that the chemist can already decide not to do something, or to identify alternative chemistry. In this paper from earlier this year, you see for dichloromethane, a problematic solvent, the toxicity space, the chemical similarity space, and the overlap. You can simply say in the green clouds, you should look for alternative chemistry, not in the red clouds. And this is showing you what you can do. Another area we are very excited about is the exposome. Something crazy, all exposure be considered. It is AI which will make this possible, and we call this exposome intelligence, EI, because it is making sense of diverse information in this field. And we are trying at the moment to start something crazy, a human exposome moonshot. We have teamed up with 3 NIH Institute, in the lead, the NIHS with Rick Wojcik, and we are organizing for next May in Washington, a human exposome moonshot, where we want to create an equivalent to the Human Genome Project for exposomics. And we have 50 organizations already joining us. It's really highly attractive. Nicole, again, is part of the organizing committee for these. I would also like to alert you of 3 articles which are coming out in the next, in about 2 or 3 weeks, which are dealing with the aspect of validation. They're looking very much in how we can move validation forward. I like to call this next generation validation. So the first one is really conceptual. That's where this picture is taken from. It opens up a lot for AI, but it also, the second article of these is about biomarker qualification and what we can learn from this to make human relevance a more important feature of our tests and the validation process. So in essence, push mechanistic validation. And the third one is closest to, this is how we envisage the change of a biomarker-driven process for validation. And the third one is trying to use AI for the validation process. So we're saying we should identify reference compounds by AI. We should model our validation studies by AI like we do for clinical trials to make them more efficient and the likelihood of success higher. We should use AI-facilitated education to inform, train the different groups, and also then disseminate the very same methods with education packages that also later the people get the same quality of training and others. So there's a lot of AI in this, and this, by the way, received an IDEA award from the complementary challenge competition. Okay, there's more to come. Nicole and I are working on a paper on how to validate AI-based alternatives and some other topics, but you have to wait for this. This is to close. I think that AI is a disruptive technology which allows us to finally integrate with the entirety of existing knowledge. And this has a prospect of accelerating product development. If you imagine 1 day faster to registration is worth $1 million for a pharma industry company. We have seen that the first one AI product developed in 2020 already completed the clinical phase 2A this year and is moving into clinical phase 2B by in silico medicine. And in 2022, we already had 18 AI first drugs which went into human trials, class 1 trials. They all started around 2019. Yeah? This is an enormous acceleration. So I think that these technologies are really a tremendous opportunity, and I hope they're opening up also for a human exposome project as really a game changer and move away from a purely genetic view of medicine over the last 40 years to a balanced view of genetics and exposure. Thanks a lot.